Hello dear students, today I am going to introduce the metabolism to you all. I am Dr. Vishwajit Ghorpade, working as Assistant Professor in Pharmaceutics at Sanjay Ghodawat University, Kolhapur. So let us begin with Introduction to Metabolism. After completion of this video, you all will be able to define metabolism. You will be able to state the meaning of various terms which are involved in metabolism, and also you will be able to differentiate in between catabolism and anabolism. Now, what is this metabolism? Metabolism or metabolic reactions are nothing but they are the chemical reactions which deal with synthesis or breakdown of the biomolecules in the living systems. These chemical reactions usually occur in series, and that series is known as metabolic pathways. If we consider the metabolism, it is made up of two processes. One is catabolism and another is anabolism. So catabolism and anabolism together form the metabolic reactions, or together form the part of metabolism. What is this catabolism? Catabolism is nothing but it is the degradation of complex molecules into simple molecules. Let us say polysaccharides. Polysaccharides are complex molecules, and if they degrade, you will get the monosaccharides, and monosaccharides are nothing but the simple molecules. So degradation of complex molecules like polysaccharides or lipids or proteins to the simple molecules such as monosaccharides or fatty acids and glycerol or amino acids is nothing but the catabolism. On the other hand, anabolism is nothing but it is the synthesis of the complex molecules from the simple molecules. Means only if you want to synthesize polysaccharides, they will be synthesized from the simple molecules like monosaccharides. Similarly, the proteins will be formed from amino acids and lipids will be formed from fatty acids and glycerol. Now this is the diagrammatic representation of the metabolic process. Here you can see that the energy-rich complex molecules such as polysaccharides, lipids and proteins, they will get catabolized to give you energy-poor products. And during this process, the ATP and NADPH will be liberated. Now the energy-poor products are nothing but the pyruvate and acetyl coenzyme A. So polysaccharides, lipids and proteins, they get catabolized to give you simple molecules such as pyruvate and acetyl coenzyme A. And in this process, ATP and NADPH are liberated. On the other hand, if we want to form or if we want to synthesize polysaccharides, lipids and proteins from the simple molecules such as pyruvate and acetyl coenzyme A, then that process comes under anabolism. So in anabolism, the complex molecules which are energy rich will be formed from the energy poor products. Means pyruvate and acetyl coenzyme A, they will come together to form these energy rich compounds. And in this process, energy will be required, and that energy is provided by ATP and NADPH. So in catabolism, the energy is liberated in the form of ATP and NADPH, whereas in anabolism, the energy is utilized. Energy is required for the formation of energy-rich complex molecules such as polysaccharides, lipids and proteins. Now let us understand what is catabolism. Catabolism takes place in three stages. As I have previously told you that catabolism is nothing but it is the breakdown of complex molecules into simple molecules, and this thing happens in three stages. So the first stage is conversion of complex molecules into their building blocks. For example, let us say that here in stage 1 of catabolism, polysaccharides, lipids and proteins, which are the complex molecules, they will get converted into simple molecules such as monosaccharides, fatty acids and glycerol, and proteins into amino acids. So here the monosaccharides are nothing but they are the building blocks of polysaccharides, fatty acid and glycerol are building blocks of lipids, and amino acids are the building blocks of the proteins. So in the stage 1, the complex molecules are going to get converted into their respective building blocks. The second stage is nothing but formation of simple intermediates such as pyruvate and acetyl coenzyme A. So whatever building blocks are formed in stage 1, it is going to get converted. So the building blocks are going to get converted into simple intermediates. Okay, they are going to facilitate the formation of simple intermediates such as pyruvate and acetyl coenzyme A. So diagrammatically, the second stage is this, where monosaccharides, fatty acid glycerol and amino acids are going to get converted or going to help in the formation of the simple intermediates like acetyl coenzyme A. In third stage, the acetyl coenzyme A, which is formed in the second stage, is oxidized. So the third stage is final oxidation of acetyl coenzyme A. So in this diagram, the third stage comes over here, where acetyl coenzyme A, it participates in the Krebs cycle. It gets oxidized in this Krebs cycle to liberate carbon dioxide, and there is formation of NADH and FADH2. Now this NADH is nothing but it is nicotinamide adenine dinucleotide hydrogen, and FADH2 is flavin adenine dinucleotide dihydrogen. These two things are nothing but they are the coenzymes, and these two coenzymes usually participate in electron transport chain or electron transfer chain, where they lead to the formation of ATPs. For example, NADH, if it participates in electron transport chain, it will liberate 2.5 ATPs, whereas FADH2, it participates in the ETC, the electron transport chain, to liberate 1.5 ATPs. So this nicotinamide adenine dinucleotide structure has been shown over here. You will see that this entire structure is composed of a molecule of nicotinamide, a molecule of adenine, and this molecule of nicotinamide and adenine are attached to the cyclic ribose sugar, and this cyclic ribose sugar molecules are further attached to the phosphate groups, where these two phosphate groups are further linked to an oxygen atom. So this is the total structure of nicotinamide adenine dinucleotide. Now we are going to call it as dinucleotide because this nicotinamide, this ribose sugar and a phosphate group together constitute a nucleotide. So in this structure there are two nucleotides. The first nucleotide, which is composed of nicotinamide, ribose sugar and phosphate group, and the second dinucleotide is composed of adenine, ribose sugar and phosphate group. Now this nicotinamide adenine dinucleotide can exist in two forms. Okay, the first form is the oxidized form, to which we indicate as NAD+. So NAD+ is the oxidized form of nicotinamide adenine dinucleotide. Here you will see that the nitrogen in the ring structure of nicotinamide is having positive charge, means there is loss of electron in case of this nicotinamide molecule, and that is why it gains a positive charge. So this is the oxidized structure of NAD. Upon reduction of this NAD, one hydrogen gets attached to the reactive site of nicotinamide over here. So this structure towards right hand side is nothing but it is the reduced structure of nicotinamide adenine dinucleotide, and we indicate it as NADH. So below you can see the total redox reaction, where the oxidized NAD+, it accepts the proton and the electrons over here. It is going to accept the electrons, and you are going to get the reduced form of nicotinamide adenine dinucleotide, that is NADH, and one proton will stay as it is. Flavin adenine dinucleotide is having somewhat similar structure, but there is one difference. The difference is that instead of nicotinamide, you will find a flavin molecule attached over here. Secondly, this flavin molecule, it is attached to a acyclic ribose sugar. Now here you will see that above the adenine molecule, it is attached to the cyclic ribose sugar, but flavin molecule, it is attached to the acyclic ribose sugar over here. And then this cyclic ribose sugar and acyclic ribose sugar, they are attached to the phosphate groups which are further linked to the oxygen atom. So this is the difference in the structure of NAD and FAD. As similar to NAD, FAD also exists in two forms, that is oxidized form and the reduced form. Now, what is anabolism? In case of anabolism, we have seen that anabolism is nothing but it is the synthesis of complex molecules from the simple molecules, and these simple molecules act as starting material for the formation of the complex molecules, and these simple molecules include pyruvate, acetyl coenzyme A, or the intermediates of Krebs cycle or the citric acid cycle. Means in this Krebs cycle, whichever intermediate moieties are formed during the entire process, these intermediate moieties are going to act as starting material for the formation of complex molecules. And as I have told you previously, anabolism process requires energy. So it is dependent on energy which is provided by ATP or GTP and the reducing equivalents such as NADPH plus proton. So what is this NADPH? So nicotinamide adenine dinucleotide phosphate. The structure is somewhat similar to your nicotinamide adenine dinucleotide, only the difference is to the ribose sugar which is attached to the adenine, it is also attached to a phosphate group. So the structure is similar to NAD, only one phosphate group is attached to the ribose sugar attached to the adenine. Now, what is the role of this nicotinamide adenine dinucleotide phosphate? Its role is nucleic acid and lipid synthesis. Okay, it is going to help in nucleic acid and lipid synthesis. It is going to help to provide energy for synthesis of lipids and nucleic acids. Okay, so remember the difference. NAD doesn't contain phosphate group attached to the ribose sugar associated with adenine, but in NADPH, the phosphate group will be attached to the ribose sugar which is associated with the adenine molecule. So with this, you have come to know that what is metabolism, what are the terms involved in the metabolism. In our next lecture, we will see what is glycolysis. Thank you.